Hello, students. Now we move on to lesson number 3, where we're going to talk about lipoproteins and hypercholesterolemia. Lipoproteins are the HDL, LDL, and the VLDL, and then hypercholesterolemia just means high cholesterol. Hyper meaning high, and then cholesterolemia meaning cholesterol. So what we're going to talk about today is how the body makes these and the implications in atherosclerotic buildup. So here's the overview of the digestive process, just to give everybody up to speed. You may have had this in anatomy and physiology, but just so everyone has an idea of how food is digested, let's talk about this. We have, when you eat, it goes through your mouth, down your esophagus, and it goes into your stomach, and where it gets digested into chyme. That chyme then gets deposited into your small intestine, where in the small intestine is where a lot of the absorption of nutrients occurs. So a lot of carbohydrates, fats, and proteins. And then moves out of the small intestine and go into the large intestine, and from there that's where a lot of water is reabsorbed, and then finally out or through defecation. Well, our focus is going to be mainly on fat digestion here in the small intestine, liver, and gallbladder. And we're going to talk more about that. So really the idea is that we're going to take what you've eaten, a fatty, high-fat food, and how that ends up becoming LDL and VLDL, as well as cholesterol. So that's where our focus is going to be, largely here in the small intestine. Before we do that, there's a couple of videos we're going to do just as an overview. So we're going to do that now. All right, so here's the video over the organs of digestion. This hopefully will be a review.
The primary functions of the digestive system are the breakdown of food, called digestion, and absorption of nutrients. Digestion begins in the mouth, where the teeth break food into smaller particles during mastication. Salivary glands located near the oral cavity secrete saliva, which begins chemical digestion and keeps the food moist. As food is swallowed, the soft palate blocks the upper pharynx to prevent food from entering the nasal cavity, and multiple voluntary muscles in the face, neck, and tongue contract, pushing food particles through the pharynx. The food passes over the epiglottis, which prevents food entry into the respiratory system, and then into the esophagus, which connects the pharynx to the stomach. The one-way movement of the food mass, now called a bolus, is controlled by wave-like involuntary muscle contractions. This movement is known as peristalsis. The bolus now enters the stomach. Folds in the stomach wall, called rugae, allow for expansion as the stomach fills. Stomach cells secrete hydrochloric acid, pepsinogen, and various regulatory hormones that chemically digest the bolus. Muscular contractions in the stomach churn its contents to further break down the bolus and mix it with stomach secretions to form a thick liquid called chyme. Chyme exits the stomach through the pyloric sphincter and enters the small intestine, the major site of nutrient absorption. The small intestine consists of 3 parts: the duodenum, jejunum, and ileum. Bile from the liver and digestive enzymes from the pancreas empty into the duodenum to aid in digestion. Absorbed nutrients pass from the lumen of the small intestine into blood and lymph. Chyme not absorbed in the small intestine enters the large intestine. As it passes through the cecum and ascending, transverse, descending, and sigmoid colon, water and salts are absorbed and chyme is converted into feces. The rectum stores feces until nervous stimulation initiates the defecation reflex, resulting in elimination through the anal canal. Chymes from the pancreas.
All right, so this is going to be very important when we're looking at the liver here. We have the gallbladder and we have the pancreas. These 3 organs are going to be very important in terms of fat digestion. And so in the subsequent slides, we're going to talk about these three and how they assist with the breakdown of the fat that we eat to eventually form the LDLs that we think of that are circulating in the blood. So just keep those in mind as we move forward. In just a moment, we're going to be watching another video about the digestion of fat, where it gets broken down. And when we talk about fat, this slide depicts what's called a triglyceride. A triglyceride has a glycerol backbone, and then it has 1, 2, and 3 fatty acids. And together they form a triglyceride. And it's these triglycerides in which the pancreatic lipases, anything that ends in a aces, is an enzyme that breaks something down. In this case, it's a lipase from the pancreas that starts to digest or break or tear away these fatty acids from the glycerol backbone. And then, so when one is taken away and you're left with 2 attached to the glycerol, that's called a diglycerol. And then when you take 2 of these fatty acids away from the glycerol backbone, it's called a monoglyceride. And so the idea is that the pancreatic lipases will come in and then they will chew this apart or take it apart. So the next video we're going to show, watch, is they're talking about the breakdown of this triglyceride, and then we'll talk about it a little bit more. So just let's show up this video in just a minute. All right, so this video is about the digestion and absorption of fats. When they're talking about fats, in our case, we're going to talk about triglycerides. So, and then once the video is finished, we'll talk more about it in the subsequent slides.
Fat digestion begins when chyme, an acidic fluid mixture of gastric juice and partly digested food, passes from the stomach into the initial section of the small intestine, the duodenum. Despite its name, the inner surface area of the small intestine is enormous. This is due partly to its great length, over 6 meters in humans, but principally to the numerous folds and tiny villi and microvilli that line its inner surface. Digestion requires many specialized enzymes as well as other secretions. The liver synthesizes bile, which aids in digesting lipids. The bile is stored in the gallbladder until it's needed to assist in fat digestion. When the chyme enters the duodenum, a hormonal signal causes the walls of the gallbladder to contract rhythmically, squeezing the bile into the common bile duct and down into the duodenum. The pancreas lies just beneath the stomach. It produces a host of digestive enzymes, including lipases, which are essential in fat digestion. The pancreas also secretes bicarbonate ions, which help to neutralize the pH of the chyme entering from the stomach. This is essential because intestinal enzymes, unlike stomach enzymes, function best at a neutral or slightly alkaline pH. Let's examine the process of fat digestion in greater detail. Fats are hydrophobic molecules, that is, they are not soluble in water. However, most enzymes, lipase included, are water-soluble and require an aqueous medium to function. Left to themselves, fat molecules would aggregate to form large globules that would present only a small surface area where the water-soluble lipases could act. Bile prevents this from happening by emulsifying the fat. The bile molecules have a hydrophobic end, which is absorbed into the fat droplet, and a hydrophilic end, which sticks out. This hydrophilic coating prevents the fat droplets from aggregating. These stabilized tiny droplets of fat are called micelles. Once stabilized in micelles, digestive enzymes from the pancreas can begin their work. Lipase breaks down the fats into fatty acids and monoglycerides. Because these products are lipid-soluble, they can easily pass through the plasma membrane in the intestinal mucosa and enter the epithelial cells. Once inside the cells, the fatty acids and monoglycerides enter the endoplasmic reticulum, where they're resynthesized into triglycerides, combined with cholesterol and phospholipids, and coated with a protein to form chylomicrons. The protein coat serves to make the chylomicrons water-soluble and to facilitate exocytosis. Chylomicrons serve as a shuttle to transport lipids through the body. The chylomicrons leave the mucosal cells via exocytosis and enter lymphatic vessels in the submucosa. From here, they're transported to the thoracic duct where they enter the bloodstream.
Okay, so there's a lot talked about in terms of the fat digestion that we'll go over in the subsequent slides. Some of the things that you just need to be aware of that we'll talk about here is we have here again is our liver. We have our gallbladder, which is going to secrete bile. And then bile, as we'll learn, is a substance that's used to take a large part of that chyme that comes from your stomach, and it basically breaks it into smaller droplets. So think of it as chyme as a big basketball size worth of fats, and bile will come in and it will break it into like the size of a golf ball. And by doing so, what you've done is you've increased the surface area for those pancreatic lipases to come in and chew it apart. So as it showed in the video here, is you have, upon eating, the chyme will come into the small intestine. So here you have, looking back, so you have the chyme that's coming in, and as a result, what ends up happening is now the gallbladder will contract, and it's going to be releasing the bile. And bile is going to be made in between meals from the liver, and it's going to be stored in the gallbladder, and then upon stimulation, it's going to be released into the small intestine. And there, that's when you have the bile is going to take this large piece of chyme, and it's going to break it into smaller pieces, as you see here. It goes from one big piece into a bunch of smaller pieces. And the idea now is it allows for an increased amount of surface area for the pancreatic lipases, depicted here in blue, to come in and break it apart. So again, just reviewing this video, moving this forward, here's our lipid droplet. Think of that as our chyme coming from from the stomach into the small intestine. And then we have there's our lipase enzyme from the pancreas. And here's our bile. And here's the bile that's made in the liver and stored in the gallbladder. And the idea is, you can see in this video what happens is now the bile is forming, and you can see what happens here. It goes from a large big droplet with not a lot of surface area into smaller droplets with much more surface area. And that allows then the pancreatic lipase enzymes to come in and to break apart the triglycerides that are stored inside of the micelle. And then again, this is a formation when you have the bile that's wrapped around the lipids, that's called a micelle. Okay, so here's the micelle, and what comes in is the lipase will come in and it'll chew it apart. And by doing so, what it does is it breaks that triglyceride down into the monoglyceride and free fatty acids, as we depicted previously. So again, this is just a picture of the fat digestion. We have our liver, here's our gallbladder, where you have the storage of the bile. And then upon eating, once the chyme comes into the small intestine, that's when what happens is the gallbladder contracts, squeezes, and that ejects bile into the small intestine, and then that's where it's going to form a micelle. And so this is a picture of a micelle where we have in the green, this is the bile. Inside are the triglycerides, and the idea now is it allows for a much easier increased amount of surface area for the pancreatic lipases, depicted in red, to come in and to break apart the triglycerides. And again, you're just tearing apart. What you're doing with the lipases is you're tearing apart off these free fatty acids, 1, 2, and you're leaving a monoglyceride. So you have a couple of free fatty acids circulating in the small intestine, along with monoglyceride, which is a glycerol with just one fatty acid attached. Again, this is just an overview of the composition of bile. Again, it's going to be dark green, yellowish brown fluid. Again, it's made in the liver, and it's stored in the gallbladder. And bile salts are derivatives or precursors that emulsify fat, which basically, like, emulsify fat is just a fancy word for takes a big droplet like the size of a basketball, and it basically allows it to be broken down into smaller, like golf ball size, to increase surface area. It facilitates fat and cholesterol absorption, which we'll see in just a little bit, and helps solubilize or change the structure of something so it is easier to dissolve, such as our cholesterol that we'll see in a minute. So not only are triglycerides and fatty acids are coming along, but also cholesterol is coming along with the triglycerides. And in just a minute, we're going to talk about enterohepatic circulation, which is the recycling of the bile salts. And this is basically where the liver and the small intestine work in between meals or right after a meal to help break down that triglyceride into the fatty acids and monoglycerides. All right, so this is the regulation of bile. Again, looking at this, there's a couple of enzymes or hormones you should be aware of. We have cholecystokinin, CCK. It's a hormone of the GI system, and it's responsible for stimulating the digestion of fat and protein. It's usually stored right here on the wall of the small intestine. You also have secretin. It's a hormone produced by the duodenum that enhances the effects of CCK by stimulating the secretion of digestive enzymes and bile from the pancreas. And here's our pancreas and also the gallbladder. So the idea is that the CCK and secretin work together to help with the breakdown of the triglycerides, or all this chyme that is now made its way into the small intestine. And so how this works is upon eating the chyme, so looking at this, the chyme enters the small intestine. Again, chyme is acidic, contains triglycerides, and again it enters the duodenum, and it causes, once it passes along the wall of the small intestine in these enteroendocrine cells, secretin and CCK are going to be released into the blood. So CCK and secretin enter the bloodstream. What they do is they circulate into the throughout the body, and they make their way back to the liver and the gallbladder. First thing that happens is the bile salts and secretin transported via the bloodstream are going to stimulate the liver to produce bile more rapidly. So the idea is that you've just eaten food, CCK, secretin are released. There's some bile salts from a previous digestive process. They're going to, CCK and secretin and bile are going to make their way back to the liver, and the liver is going to use the bile salts to make new bile. At the same time, you have a vagal stimulation from the brain that's going to cause some weak contractions of the gallbladder to kind of prime the pump, so to speak. And then finally, you have CCK via the bloodstream is going to cause the gallbladder to contract more forcefully. And the idea now is that bile is going to make its way down to the sphincter, here into this hepatopancreatic sphincter, hepato from the liver, pancreas from the pancreas. And what's going to happen is that bile is going to travel down to the sphincter, and it's going to be released, bile is going to be released into the duodenum. And then that bile then is going to do what we just showed here, is it's going to take the triglycerides that are within that chyme, and it's going to make that big droplet of chyme into much smaller, like golf ball size. So taking a basketball size of chyme and making it in a bunch of small size golf balls. Meanwhile, the pancreas is going to release pancreatic lipases, and the lipases are going to come in and start to break apart the triglyceride. So this again is just what happens after the regulation of bile release after you've had a meal. And again, we're going to focus our attention with fat. So this is the how bile is released and the role of bile in the pancreatic lipases. So all we're really talking about so far is how the food you've eaten has made its way into the stomach, and it's been broken down into chyme, and then through the use of bile, it's going to be formed into smaller droplets called micelles, and then the pancreatic lipases will break apart, attack the micelle, and break apart the triglyceride into fatty acids and the monoglycerides. and they're going to remain here in the small intestine. So you should pause the video here, take a few moments, look over this, make sure you understand the 6 steps of the release of bile after a meal, because this is the first step in the digestion of fats that eventually lead to the lipoproteins, LDL, that we talk about as the bad cholesterol. So take a moment, pause the video, and make sure that you have a good understanding of how the release of bile and the digestion of micelles and the triglycerides. All right, so again, just to review, here we have, just to review where this is all occurring here in the absorption in the small intestine. It's happening in the lumen, which is the opening of the small intestine. And so this is where you have the micelles that are being broken down by the pancreatic lipases. Next up, what we're going to talk about in just a little bit is the absorption, how you get those monoglycerides, cholesterol, and fatty acids from the lumen of the small intestine, and how they eventually make its way into our circulatory system or the blood. First, though, let's take a moment and review these questions. So let's pause the video here, making sure that you answer each of these questions. Take a piece of paper out and answer these questions, and then we will review in just a moment. All right, so welcome back. Assuming that you've taken a moment to review answer these questions to have a good foundation of what we've talked about so far. Let's answer these. So where is bile made? It's made in the liver, mainly in between meals. Where is bile stored? It's stored in the gallbladder. And micelles are composed of what? Bile and triglycerides. Again, the micelle is used to take that big chyme that comes right out of the stomach and the small intestine, and it makes it from a basketball size, so to speak, into much smaller golf ball size. And by doing so, you've increased the surface area for the pancreatic lipases to break down the triglycerides. What are bile's 3 functions? To emulsify fat, okay, facilitate fat and cholesterol absorption, and then help solubilize cholesterol so it can be digested. So essentially, the idea that bile is used to help with the facilitation of taking the fat that we've eaten and breaking that down and allowing for the triglycerides to be broken down into monoglycerides and fatty acids to be digested. And again, cholesterol is also coming along with the monoglycerides and the fatty acids. What hormones stimulate the digestion of fat and protein? Again, CCK, cholecystokinin, and secretin, as we just saw in the circulation and the release of them from right after a meal once they chyme makes its way into the small intestine. And then fat absorption occurs in the small intestine. Okay, so this is hopefully an overview. Make sure if you have that you have a good foundation. If not, take a moment, review the notes, the slides so far, or watch the video a second time. Because now what we're going to move on to is what happens, how does it get the fatty acids and the monoglycerides and the cholesterol, how does it go from the lumen of the small intestine and eventually makes its way into the blood? Because that is then the precursors to the LDLs that we think of as the bad cholesterol, so all the lipoproteins. Okay, so at this point, we'll end this section of the video. Make sure you review to have a good understanding.